So it's my great pleasure to present you my work on small RNA sequencing. So this is just bioinformatics, but I promise you that I won't have any equation or algorithms. So I just wanted to present you some of the tools that have been developing for the last few years now. So I will start with a very simple pipeline that you may use when you sequence your small RNA. Basically, you usually start with sequencing, and afterwards you usually, most of the time, let's say, align your reads to the genome. And then two possibilities: either you just have, let's say, one sequencing data, only one data set, and you want to quantify your small RNA classes, for instance, microRNAs and so on and so forth, or you want to compare two data sets, and you want to do the test for differential expression. So these are the, let's say, main step of the analysis, or the first step, let's say. So I just wanted to present you my contribution on these three tasks. So the first one is the genome alignment. I guess you may use these tools, for instance, bowtie, BWA, HiSat, STAR, and so on. So they are very efficient tools and they work fine, but still they use heuristics, meaning that they won't necessarily provide you all the hits with the minimal, let's say, number of mismatches. They might miss hits or give you something which is suboptimal in very few cases, but sometimes it happens. And sometimes they also limit the number of hits that you may have. For instance, they will give you one possible location, but not all the locations. So this might be a problem, for instance, if you're studying mismatches, let's say, RNA additions that may happen at the ends, let's say, of microRNAs. And it is known that they are not optimally handled by all these tools there. And also, second is we will see also that afterwards that small RNAs are often duplicated in the genome. Think about piRNAs or silencing RNAs that you can, it's very hard to find a precise location of these RNAs on the genome. So you oftentimes have many hits for one read. So I just wanted to mention sRNA mapper, which is, it has been submitted to a journal, but not yet accepted, but still on the benchmark. So it works better than the other known tools, meaning that it provides all the best hits. And it's usually run faster than most of the other tools. And it's run even faster if you provide several FASTQ files that sRNA mapper map all at once, actually. So this is for the first tool. Second tool is when you want to quantify, for instance, your RNA, your small RNA classes. You sometimes want to have these pie charts that you would like to produce, let's say, and you want to quantify the number of tRNAs that you have, microRNAs, and so on and so forth. The problem here, as I said previously, is that you have microRNAs which are often duplicated in the genome. Small RNAs which are linked to repeats are repeated, obviously, and the same goes for tRNAs. So let me show you a short example here. For instance, when you have one read, A, which maps only once, so you want to say that this read is upstream, but B and B here are, these reads can map at two different locations. But these B, they both co-localize with microRNAs. So in this case, you want to say that even though B maps at two different places, you are pretty sure that it comes from microRNA. And this kind of ideas is used by MMANOT to consider all the hits of your RNAs, of your sequencing RNA, to try to find the best, let's say, annotation for all the reads so that you can compute the pie chart that I've shown you previously. And so this work has been published, and it has been shown to map or to use, let's say, more reads for the annotation. And if you compare it to another strategy, which is you want to restrict to unique matches, well, it works better this way, because when you restrict to unique matches, you just consider a very small subset, actually, let's say half of your reads. And so the repertoire that you consider is really restricted. So this is MM-ANNOT, and if you want to consider the expression of simple genes, let's say, not classes of genes, then you may want to use MM-QUANT, which does a similar job. Last tool is applied to differential expression. So the aim is quite clear. You want to find all the differentially expressed genes. So what you usually do is you count the number of reads that's co-localized with your annotation. And afterwards, you compare the number of reads and you do the test, right? But the thing is that, I mean, this happened to me, and I guess that it happened to all of you, is that you got a lot of reads which do not co-localize with anything which is known. And what you would like to consider is what are these things which are differentially expressed, but you don't exactly know what they are. You just want to look for everything which is differentially expressed, whether or not it co-localizes with something which is annotated or known. So this is what RNA diff does. It uses your BAM files, and afterwards it uses 3 different strategies that I call HMM, RR, and NAIVE to find several regions. And all these regions are merged together to give you a list of differentially expressed regions. Why 3 different methods? Because sometimes you have reads which are in, let's say, large, genes, like pi-RNA, for instance, which are in a very long transcript. Or sometimes you have something which is very sharp, just like microRNA. So this is why you may want to use several different strategies to find everything which is differentially expressed. Well, this is it. So this is the summary slide. So you can use sRNA mapper to map your reads, and afterwards for the quantification, MM-ANNOT to quantify the classes of small RNAs, and MM-QUANT to quantify their genes. And for the differential expression, you can use sRNA diff. So sRNA diff, for instance, is available on Bioconductor, and the other tools are usually available on Linux or whatever, as long as you can compile the source code, which is written in C or C++ every time. This is it. So I'm happy to take questions if you have.